And we do have a biotechnology test today, so here are a few notes that you might want to jot down before that test. We talked about the process of taking the plasmid, which is the circular DNA from a bacteria, and splicing it or cutting it open and putting in a human gene, and then it closes back up, and then the bacteria will reproduce carrying that human gene and able to produce, one of the cases we talked about was produce human insulin. Okay, so that process can go by several different names. So we have things like gene splicing. And that's gene splicing because you're cutting the gene open. It's also called genetic engineering. It can also be called recombinant DNA technology. These all refer to the same process: genetic splicing, gene splicing, genetic engineering, recombinant DNA technology. They all refer to altering the genes of an organism or the genetic makeup. In order to get a desired trait. And that's the whole reason for doing genetic engineering, gene splicing, recombinant DNA technology. The whole reason for doing that is to get an organism with a desired trait. So we're changing the genetic makeup of an organism in order to get a desired trait. And when you do take genes from one organism and put it into a different organism, a different species, a different type, like from a human into a bacteria, okay, or from a fish into a tomato plant, any number of those. If you take genes from one organism and put them into a totally different organism, then that is called transgenic. Trans means a cross. You're taking a cross from one organism into another organism. So it's transgenic. It's a transgenic organism, and it's when you take genes from one organism and put it into a totally different organism. It produces a transgenic organism that has genes from somewhere else. Okay, when a farmer or a scientist has an organism that has the trait they want, maybe they've produced a goat that can make high protein milk or milk with a certain protein in it, and they want to make more just exactly like that organism they have, they do what's called cloning. Okay, so cloning takes the genes from an organism with a desired trait. You're going to remove the nucleus from the egg of a surrogate, and then they're going to take a nucleus from a somatic cell of that donor, the one that has the desired traits. They're going to take a nucleus from one of the body cells of that organism that they have that they like. And they're going to implant that nucleus into the egg of what's going to be the surrogate mother. So they'll plant that nucleus into the egg. That egg's not going to have to be fertilized because it has a somatic cell nucleus in it. So it already has the complete set of chromosomes. Because if it were fertilized otherwise, then it would not be genetically identical. But in cloning, when you want to make an organism that is genetically identical to another organism, you take the nucleus from a somatic cell and plant it into the egg of the surrogate. And then it starts to divide. It makes a zygote. That zygote would then be implanted into that surrogate mother there, and she would carry that offspring and give birth to a clone. The offspring would be genetically identical, not to the surrogate mother that gave birth to it, but genetically identical to the clone or to the original organism that donated that nucleus. Okay? And that's called cloning, and it's a way to make organisms that are genetically identical to another organism. So when you use the genetic information from a single parent organism to produce offspring that are genetically identical to that parent organism, it's called cloning. And the genetically identical offspring is called a clone. They're genetically identical to the one that donated that nucleus. And cloning is based on the idea from the cell theory that all cells come from other living cells by cell division. So if you make a cell that has a certain genetic makeup, then it's going to divide, and those cells produced when it divides are also going to have the identical genetic makeup. So that all cells come from other living cells by cell division allows cloning to work. We also talked about DNA fingerprinting. DNA fingerprints are produced by using restriction enzymes to cut a piece of DNA at specific locations. The DNA fragments are then put into the wells of electrophoresis gel tray. Those DNA fragments are negatively charged. You might see that. They're negatively charged due to those phosphate groups. This causes them to move toward the positive end of the gel when an electric current is applied. The smaller fragments will move farther down the gel. Okay. It's like two people trying to squeeze through something. A smaller person is going to be able to squeeze through faster than a larger person. So the smaller fragments are going to move farther down the gel. Okay. Important to remember, no two individuals have the same DNA fingerprint except for identical twins. Okay, and that's the difference between DNA fingerprints and regular fingerprints. Nobody, even identical twins, do not have the same fingerprints. Okay. Another difference would be fingerprints are only found on the fingers. DNA is found in every cell of your body, so your DNA fingerprint can be found from any cell. If you use a skin cell that might get left behind at a crime scene, or a drop of blood that might get left behind at a crime scene, can be used to identify someone who was actually there at the crime scene. So a DNA fingerprint is present in every one of your cells. Okay. They can also use not just to identify someone present at a crime scene, it can also be used to identify the parents of the child or the child of a parent. It can help narrow down who may or may not be the parent of a child. Genetic fingerprinting can be, like we just said, using in forensics. Forensics is using science to solve crimes. So in forensics, it can be used to place a criminal on the scene of a crime. You would take the evidence and you would find the suspect. Its DNA fingerprint would match up with the DNA fingerprint found at the crime scene. Okay. If you're identifying parents of a child or a child that might belong to a parent, remember the child is going to get DNA from the mom and the dad. So the child would have bands that would match up with mom and bands that would match up with dad. If there's a band that doesn't match up with either one of them, then that would not be the child of those parents, because the bands that the child will have will either came from the mother or the father. So when you look at mom and dad, the bands on that child are going to be either in one or the other's DNA. And again, identical twins are going to match exactly. So if you're looking at DNA fingerprints and comparing them, if there are two that match exactly, then they could be identical twins, but they would have to match exactly to be identical twins. Okay, we also talked about gene therapy. Gene therapy is a really, really cool idea, and they're seeing some success with it. It is still in the experimental stages, but lots of disorders, such as cystic fibrosis, hemophilia, sickle cell anemia, a lot of those disorders are caused by a gene that is either missing from the DNA, or that we talked about mutations, about sometimes bases can get arranged in the wrong order. Maybe an incorrect gene, a gene that has a mutation in it that causes it to not produce the correct protein. The idea behind gene therapy is to place a corrected gene into the cells. Okay. So if someone is missing a cell that produces the enzyme in cystic fibrosis, if they're missing that gene that produces the enzyme needed to break down the mucus, and as in cystic fibrosis, then that gene, theoretically, they can take that gene and implant it into cells, and as those cells divide, it makes more cells that have the gene, and the body starts making that protein that it needs. And that's called gene therapy, and it's a up-and-coming branch of medical science. It's a really awesome idea. It's seeing some success. Hopes to see lots more. One place they're using it again is with muscular dystrophies. Certain muscular dystrophies, they're using gene therapy. And the idea is, we talked about viruses and how viruses are able to attach to a cell and then implant their genetic material into the genetic material of that cell. So the idea behind gene therapy, a lot of times they are using, again, a virus, because a virus makes a good vector for carrying a gene into a cell. They are able to attach to a cell and insert their genetic information. So for gene therapy, they can take the desired gene and put it into a virus and then use that virus to transmit that gene into the cells. They tried for years, like in vitro, in the lab, to put it in there, but when you use a virus, it can go to more cells at one time, and viruses are really good at putting genetic material in the cells. So gene therapy a lot of time uses a virus to carry that desired gene and implant it into cells. Really cool idea, lots up and coming in that field. We also talked about genetically modified organisms, also known as GMOs. And we talked about some good things about it. The BT corn can produce its own pesticides, which saves the farmer from having to use so much pesticides, which again is good because we don't need the pesticides. But then it also can lead to some environmental issues that we talked about. Those pesticides may end up wiping out organisms that are necessary for the ecosystem and cause harm to the ecosystem, because everything in that ecosystem is there for a reason. It all has its niche and serves its purpose. So when we start messing around with the genetic makeup of these organisms and introducing them back into the ecosystem, we're not sure how that's going to affect the ecosystem over time. So there's things like BT corn that would be a GMO. Golden rice we talked about. Golden rice is very important to the diet, especially in like third world countries where they rely mostly on rice, and that's the main staple of their food. They don't have a lot of other things to eat. A lot of children were going blind because rice on its own is not very nutritious. And they made golden rice, which has been genetically modified to produce provitamin A, and that helps to prevent blindness in those children. Genetically modified plants can improve nutrition and increase the plant's likelihood of surviving and living to maturity. This can increase crop yield, which is really good for the farmer, really good for those of us who like to eat, because we can get more food at a lower price if it has a better yield. But then there are those ethical and those environmental issues. GMOs tend to be produced in only one variety of, if it's the GMO corn, maybe they use just one variety of corn. Each farmer may have his own seed that he was grown for years, and his neighbor next door to have his own that he's grown for years. And you have several different varieties in an area. So if a disease comes through and wipes out one farmer's corn, then the other farmer's corn might be resistant to that disease, and so there would still be corn available. When you reduce that genetic variety and everybody's using the exact same kind of corn or exact same kind of wheat or whatever it might be, everybody's using the exact same thing and a disease comes through and that corn or wheat's susceptible to that disease, then all of that corn or wheat's going to be susceptible. And it's going to wipe out the entire crop, maybe for hundreds of miles even. So genetic variety is important, and using GMOs a lot of times reduces that genetic variety. We also do not know how those GMO organisms are going to affect the other organisms in the ecosystem. Are those pesticides going to end up wiping out animals that are important, maybe pollinators or animals that are important in the ecosystem for survival? Are those crops that are made to be more resistant to herbicides so that the farmer can spray the whole crop with herbicide and kills the weeds but not the plant? Well, there was some fear that those herbicide-resistant plants might end up fertilizing plants in the wild, and then you have plants that become weeds that you can't treat with herbicides because they're resistant. So, again, you don't know how those GMOs are going to affect the ecosystem. And then you have things like Monsanto, who has formed a monopoly. That's a company that makes seeds, produces genetically modified seeds, pesticides, herbicides. They kind of have a monopoly on all that farming stuff up there. And they have increased genetic diversity. They're pretty much forcing farmers to use their corn, their seed, and they won't allow them to bring in others. So Monsanto has formed a monopoly out there, and that's another danger with the GMOs. Okay, so that's some things that you might want to jot down and make sure you remember before we go into the test in just a little bit. Good luck.